Hello, everyone, and welcome to Brain Math. We are very glad to have you here, our Zoom audience too. So without any further ado, it is my great pleasure and honor to introduce our speaker today, Professor Earl Miller. Earl Miller is the Pickaware Professor of Neuroscience at the Massachusetts Institute of Technology. He has an academic appointment at the Pickaware Institute for Learning and Memory and at the Department of Brain and Cognitive Sciences at MIT. So Professor Miller earned his BA with honors from Kent State University in 1985 and his MA and PhD from Princeton University. In 2020, he received an honorary Doctor of Science degree from Kent State University. Professor Miller's work has been pivotal in understanding the neural basis of cognition. He has made significant discoveries in areas such as working memory, attention, categorization, and top-down executive control. He developed a key theory in cognitive neuroscience, emphasizing the role of the prefrontal cortex in rule learning and goal maintenance. Professor Miller pioneered the study of multifunctional mixed selectivity neurons, making a departure from traditional views. He has provided new insights into the role of neural dynamics in shaping perception, thought, and action. By combining experimental, theoretical, and computational methods, his work offers vital insights for understanding cognition and its disorders in conditions such as autism, schizophrenia, and attentional deficit disorder. Professor Miller has received numerous awards and serves in editorial roles for major neuroscience journals. He also serves on international advisory boards. His 2001 paper with Jonathan Cohen, introducing a novel theory for understanding executive brain functions, is the fifth most cited paper in the history of neuroscience. So with that, and reminding everyone that this seminar is co-sponsored by the P41 Center for Mesoscale Mapping housed in Martinos. I will pass it to you, Earl, and thank you so much for being with us today here. Thank you.
Thank you, Abbas, and thank you for the invitation. It's my honor to be here. Hi, everybody. Thanks for coming out and hearing me speak. I'll just get right started. So this is our laboratory, and what we do is we study executive brain functions. These are the brain functions involved in planning, focusing attention, learning and following rules, organizing thoughts, goal-directed behavior. And we study them by recording electrical signals from the brains of non-human primates. And we use computational modeling and analytics to make sense of these signals. And why do we do this? Because we want to understand cognition. Understanding the brain is cool, it's fun, it's a great thing to do. But we also want to understand brain dysfunction diseases so it can open up new therapeutic paths to addressing diseases like autism, schizophrenia, and ADD. So our main, we use multiple electrode recording to study many areas, cortical areas, and subcortical areas in the brain. But our main interest, our fulcrum, is the prefrontal cortex. The prefrontal cortex is central to executive functions. We know it's critical for planning, monitoring, and executing complex goal-directed behavior. And our hypothesis was that the major prefrontal function must be to learn the rules of the game, of possible goals, and the means to achieve them. Before we started doing our work, and I'm not the only one to make this point, by the way, don't want to imply that, but the dominant model of prefrontal cortex function a while back was working memory. We thought, well, working memory is a good thing to have. It's important cognitive function, but the cortex, the prefrontal cortex, the executive, needs to learn how the world works. Needs to learn out what goals are available and how to get there. So we apply newish kind of technology, multiple electrode recording to the brains of non-human primates so we could understand complex network properties. And I say newish technology, it's not new anymore, but when I was a graduate student, state-of-the-art was recording from one electrode and one neuron at a time, and we were trying to figure out the brain one neuron at a time. And our knowledge about how the brain works led to more advanced thinking about brain function, and also this technology to really record from thousands of neurons simultaneously, and things like local field potentials, electric fields, allows us to now begin to study the brain on a network level in terms of its emergent properties. And I'll give you an example of emergent properties in a moment, but what they are is they're properties and mechanisms, network actions of the brain that only emerge only when the parts interact. They can't be studied one neuron or one brain area at a time. So I'll be talking about two different types of signals today. Spikes, these are action potentials of individual neurons. So think of them as the individual voices of a very large crowd. And local field potentials, which is the average activity of millions of neurons near the recording electrode. So the roar of the local crowd. So we're looking at both levels. Now, when we started our work on the prefrontal cortex and executive functions 30 years ago now, we started a series of investigations. We wanted to understand the role of the prefrontal cortex in learning the rules of the game. So we started teaching monkeys all sorts of top-down type of information, not like hold this picture in working memory. That's important because the animal's learning a task, but we also want to learn how the animal gains knowledge about the world. So we did things like we trained monkeys to recognize the categories cat versus dogs. And this is what you see here on the slide. We had 3 prototype cats, 3 prototype dogs. We morphed between them, so the shape changed smoothly between all the prototype cats and dogs. But we taught the monkeys that there was a sharp dividing line between the cats and dogs that didn't actually exist in the real world. It doesn't make a difference where you draw the line, you get the same result. You get neurons in the prefrontal cortex that are activated by whether a stimulus is a cat or a dog, and they don't really care about the individual cat or dogs. So the prefrontal cortex is learning what's important about the task and skipping over the sensory details. That's what your sensory cortex does. And we also taught other things, like taught animals to learn abstract dot patterns. Same sort of thing. We have a prototype, 2 prototype patterns, and then we make distortions of them, exemplars, the animals that learn those. And then we taught the animals when Andreas Nieder was in the laboratory. He trained monkeys to recognize the numbers 1 through 5. And what he did is he carefully made the stimuli so that there's no way to solve this task by using low-level visual features, which you see across the rows here, low visual features. And what happened was the monkeys had to learn to recognize these numbers 1 through 5, regardless of how they were presented. And we found neurons in the prefrontal cortex that activated to the numbers of 1 through 5, and they didn't care about what the display looked like. Then we took this further and we taught animals rules and concepts like same versus different, up versus down. And what we found across all these studies is that the prefrontal cortex indeed absorbs top-down information. The spiking activity of its neurons, they don't care so much. They're relatively insensitive to the sensory details, what individual cats look like, what these individual displays look like. What they do is they represent the rule, the concept that you taught the animal, what the numbers were, the cat versus dog categories, the up versus down, or same versus different. So what the prefrontal cortex can do is track the top-down information, the rules and principles the animal needed to solve the task. And then we went on to show how the prefrontal cortex can use top-down information to control the flow of activity across cortex. And this is a paper I wrote with John Cohn nearly 25 years ago now. And what we showed is that this top-down information basically provides feedback signals that feedback to the rest of the cortex and bias the flow of activity in posterior cortex in ways that match this top-down information, in ways that match the goal and the rules the animal use to solve the task. The general idea is the prefrontal cortex is like an orchestra conductor. All this top-down information is like the prefrontal cortex learning the score, then it directs the orchestra being the posterior cortex. Okay, but in doing these studies, we discovered something new. Now, when I was a graduate student, we all used to think that cortical neurons were relatively simple and specialized for single functions. You guys have probably all heard of the simple and complex cells, the human and weasel neurons that respond to an edge, or some neurons will respond to a corner. And that was the idea. That's what we thought of the brain. We thought that all the neurons in cortex, every neuron had a single function. And what they would do, they would detect simple features like edges, or they might combine a few features into one percept. Like you might have a neuron that combines, the color red and the round shape here and the motion to contribute to the percept of a red rolling ball. So that's traditional selectivity. That's the way we used to thought the brain worked. But now we start teaching the animals for the first time all this high-level top-down information and recording from hundreds or thousands of neurons. So we're not pre-selecting neurons for study. We're studying them in an unbiased fashion, getting a real picture of what the population is like. We found another population of neurons, which we eventually called mixed selectivity. And these are neurons that did not fit this traditional mode. They didn't seem to have one function. Instead, these neurons were selected for complex, nonlinear combinations of different features of the task that seemed to have no obvious relationship to one another. And they were multifunctional. If you have the same neuron, you can track the same neuron while the monkey switches between different tasks, and you'll see the same neuron basically signaling different signals in different tasks. Now, every time you go back to the same task, the neuron does the same thing, so there's consistency, but the neuron seems to be like a utility player in a baseball team. It does different functions in different tasks. So that was weird. At least at the time, it felt weird. And we did get some skepticism over this, but this is now well established in the field, these ideas, mixed selectivity, multifunctional neurons. But why are these mixed selectivity neurons so important for top-down information, complex goal-directed behavior? Well, we first called these neurons, John Duncan and I first called these neurons adaptive coding neurons about 2000 or so. And then what happened is more recently, in 2013, Stefano Fusi and Mattia Rigatti, who are computational neuroscientists at Columbia, they worked with my student, Melissa Warden, who's now a professor at Cornell, and they devised a computational model that showed why these mixed selectivity multifunctional neurons are so important. To make a long story short, they add horsepower to the brain. Your brain can't do high-level cognition without these neurons. They combine a wide range of information, which is what you need to engage in. You've got arbitrary behavior, the kind we're engaging in now. They make the brain smarter because they create a high-dimensional representational space that allows the brain to represent more complex things. And because information is distributed across many neurons instead of just a few specialized neurons, this increases the brain's storage capacity. It makes the brain essentially capacity unlimited. And for all these reasons, it contributes to the kind of things we think about. We think about high-level cognition and executive brain functions, fast learning and cognitive flexibility. Okay? So, but this mixed selectivity requires another new way of thinking. It's not just these neurons, we discover a new property of neurons. It requires a new way to think about the brain. Because the way we used to think about the brain is that every neuron has one function, as I mentioned, and anatomy is destiny. Neurons combine signals because they're wired together. So in the old Hubel-Wiesel model, two simple cells that represent two different edges will combine to a corner neuron, and then the corner neurons will combine to a complex shaped neuron, and you combine, eventually you get the neurons that do peace, love, and understanding, right? You have a lot of neurons in your brain. So it's a combinatory process, and it's all done by anatomy. It's all done by which neurons are connected and their synaptic weights. So the idea was that every perception, every thought action had an anatomically unique network, something we call an ensemble. So here I've drawn one thought. There's a blue ensemble corresponding to one thought, and there's a red ensemble corresponding to another thought, and they were separate. And you can develop associations between these thoughts. Like let's say this is a blue is a dinner bell and red is dinner. You can develop some links, some associations between ensembles, but generally they were thought to be separate anatomically unique ensembles for every thought, perception, action you have, every memory you have. But mixed selectivity means that neurons aren't members of single ensembles, they're members of multiple ensembles, multiple networks. So I've drawn this here. Here's our red and blue networks, our red and blue ensembles combined into one network that share common elements, these multifunctional mixed selectivity neurons. So how does this work? Anatomy is destiny, which is what we used to think. How does this work? If I want to activate like the red thought, if I activate those neurons, then via these connections, the activity will run through the mixed selectivity multifunctional neurons and activate the blue network. Now we have both ensembles activated, and neurons participate in many, many networks. Now your brain's a jumble of thoughts. So why don't networks all activate one another and create a jumble of thoughts? How does the brain select one ensemble from this overlapping anatomy where neurons sharing common elements? How does the brain do that if anatomy is destiny? Well, anatomy can't be destiny. The answer is that multifunctional neurons means anatomy is not destiny. The brain doesn't, mind you, connections between neurons, synaptic weights, are very important to the latent information your brain has. But your brain can't work alone by just pure anatomy because of this overlapping network problem. So neurons are members of multiple networks with changing your participation. It's not, the wiring is important, but it's not just the wiring. And cognition is a large mesoscale organization of brain processing. A simple creature, a sea slug, can swim towards light or swim towards food or swim away from a predator. That's the environment organizing that creature's brain. It's activating circuits. But we, as complex creatures, we can act on the environment. We choose what to pay attention to. We choose what to do. What that is that your brain organizing itself on a large mesoscale, coordinating the activity of millions of neurons to direct it towards whatever you decide to do at that moment. So how does the brain organize this? How does the brain produce this, get this large-scale mesoscale self-organization if anatomy isn't the full explanation? Well, insight is coming from examining the brain at a higher level, namely these brain's emergent properties. So what are emergent properties? Well, emergent properties are not explainable by individual parts alone. So here I have a video here, a GIF of a crowd doing the wave. You see the wave moving from right to left across the computer screen. Hope you can see that. And if you studied each person in this crowd one at a time, let's pretend they're single neurons. If I had a single electrode on each of these persons, or I gave each person a microphone and studied them one at a time, I would get great detail on what everybody's saying, but you would never see this wave. This wave only comes from the crowd acting together in real time, influencing one another, and that's how you get the wave. So you can't see this wave by studying each person separately, even if you studied the whole crowd. And likewise, you can't figure out how the brain works by studying the brain's single neurons or single areas at a time. So what does that do with the brain? Well, your brain has a very prominent emergent property. It oscillates. So brain waves are EEG tracings. You guys all know what this is, but your brain oscillates on a wide variety of frequencies, everything from delta, like 1 time a second, through theta, alpha, beta, gamma, 30 hertz and above. There's all these nested oscillations going on in your brain. And what that is the brain, or that's natural organization of the brain. My guess is what happened is that as nervous systems started to become more and more complicated, they started to oscillate. And that's what electrical elements do. You connect a bunch of electrical elements into a network where they can influence one another, you will get oscillations. That's what electric networks do. But the moment the nervous system started to oscillate, now you have an organization. It's a dumb organization, but neurons are now activating and deactivating together. And evolution found a way to push that around in order to create this large mesoscale organization. So just as a stadium crowd can self-organize by following a few simple rules, like stand up and the person left me stands up and sit back down again, evolution can harness these same wave dynamics to organize the brain. And plus, wave dynamics make it easier to gain mesoscale control of the brain, because the alternative is the brain somehow gates a gazillion synapses on a gazillion neurons and produces this large mesoscale of millions of neurons all coordinating their activity. But waves are relatively simple. Wave functions are relatively simple. And if you can harness those, now you can control the brain at a level which is not hopelessly complicated. Okay, but at the same time, simple rules like wave functions can actually interact to produce very complex patterns. So, and this mesoscale organization is what's needed for high-level cognition, because attention, decision-making require coordinating a large number of neurons around a particular function. Plus, in addition to this problem of mesoscale organization, there's another problem the brain has, which is representational drift. Neurons are fickle. If you record from 100 neurons under 100 identical trials, 100 identical conditions, you'll get 100 different patterns of spiking individual neurons. Some neurons will spike, then they don't spike for a while, then they spike again, then they don't spike for a while, then they spike again. So the cortex is like an orchestra of fickle musicians. There's a song is playing on, function is going on, but each neuron is playing a little bit of the melody, that they're being quiet for a while, then they're playing a little bit of the melody, that they're being quiet for a while. This is where your cortex works. Cortical neurons are not firing 80% of the time, at least. They're silent most of the time in terms of not spiking. So the whole time they're not spiking, their membrane potentials are fluctuating, electric fields around them fluctuating, and they're spreading these electrical influences to all the neurons around them. So most of the time, neurons are in a state where they're not spiking, but they are perpetuating electric fields that can influence the surrounding neurons. Okay, now, it's a representational drift problem. This was tackled by Dimitris Panossis, my collaborator from City College London. And what Dimitris did is he looked at the brains, he looked at LFPs, and he extrapolated just one step above the LFPs, the brain's neuroelectric fields. We're not talking about LFPs are very close to we're talking about the summation of LFPs that are just above the brain's infrastructure, but hover around the brain's infrastructure, just a slight averaging above the LFPs. And what he found was that you can read the contents of working memory, not by studying spiking neurons. You can do that too, but you can read the contents of working memory just by studying electric fields alone. So we had the animal hold one of 4 pictures in working memory over a memory delay, and we could actually read in real time which picture the animal is holding in working memory by using the brain's electric fields. We tried to do that by spiking activity. We had 100 neurons spiking, but neurons are too fickle. You can't do it without averaging. You can't do it in real time, and you have to do averaging. Whereas electric fields, you can read the brain in real time, read information in real time without any representational drift whatsoever. So it's just a pure informational signal. Now, doesn't that sound useful? You have a higher level of processing where there's a pure informational signal going on with no drift at all, and it's reflecting the brain's function.
Can I interrupt just for 1 second?
For 1 second, yes.
Is there a natural spatial scale to when these, say, electrical properties begin to emerge coherence? Something that probably is great and not material.
Is there a spatial scale? Yes, the spatial scale will depend on the frequency. So gamma, which is the highest frequency, local influences, even frequency and space. So the gamma is high frequency, and gamma is shown to have very local influences, which makes sense because we think gamma is what I'm going to get to this in a moment.
Electrons,
Looking at the hundreds of micron level, yes. So the level of where you expect individual ensembles to form. Then as you get lower frequency, the spatial scale gets larger and larger and larger. And for now, jumping ahead, we think that alpha beta, which is just below gamma, that's the right scale to produce organized top-down control. Then you get to lower frequencies like theta, and theta is it's very low frequency, it's 4 to 8 hertz. It's thought to be a carrier wave that helps gamma spread over long distances in the brain because gamma can't do it on its own because it's local influences. Then you get down to delta, which is like 1 hertz, and this is like general state changes in the brain. So there's a direct relation between frequency and spatial scale, and there's different, you want the contents of thought to be local and high spatial scale, low small spatial scale. Then the control has to be on a larger scale. So we think that's why lower frequencies are involved in control. Okay, but this is not just useful to study by Dimitris. It's not just useful for us studying the brain. It's useful to have this signal that's non-representative, that doesn't have any drift. But if we can use it to study the brain, then evolution can find a way to help the brain operate, right? And this is not speculation. There's something in the brain called affective coupling. Affective coupling is electric fields have a causative influence. If you go do a Google Scholar search on affective coupling, you will find thousands of papers. This is not theory. This is not in principle. This is actually happening. Electric fields have a causative influence. And the way it works is spiking activity generates local electric fields. Then the electric fields turn around and sculpt the spiking. They control the spiking. And it's got to work this way because electromagnetic influences are a two-way street. The influences are not one way. They're both ways. And this is so we think that this electric field influence is helps organize the brain on this meso large scale. But we've also proposed this could work on the micro scale too. We propose a theory called cytoelectric coupling, where electric fields we think can restructure the brain on the molecular level, the structural protein level, so to fine-tune networks to process information more efficiently, to oscillate more efficiently. And the way to think about this is I've drawn here. You guys maybe have seen this before. If you put a bunch of sand on a speaker and you play a note, the sand will organize, right, into like in this case, a circle here. Well, the electric fields do the same thing to the brain's structural proteins, can shuffle them around to make them reflect the oscillations, and that makes them work more efficiently, makes them oscillate more efficiently, makes them process information more efficiently. Kind of like tuning up the guitar string. So the way we think about this is brain anatomy is not destiny. Brain anatomy is crucial. It's important. It's where everything starts. Spiking is also where everything starts. But brain anatomy is more like the road and highway system. It doesn't say where traffic goes. It says where traffic could go. And what your thoughts are is how the traffic actually flows on this infrastructure of anatomy in your brain. In fact, we have some evidence we mounted over the past 10 years that your brain actually uses this to help partially answer your question. The brain uses different frequency bands to carry different types of signals. So we have the back of the brain, sensory cortex, front of the brain, executive cortex. We've been finding evidence that sensory information is fed forward from the back of the brain, sensory cortex, to the executive brain, frontal cortex, by using higher frequency gamma rhythms, because gamma is a small spatial scale, so it's good for it's an ensemble level kind of microorganization. And we also have considerable evidence now that beta rhythms, just below gamma rhythms, they actually flow in the opposite direction. They feed back from frontal cortex to sensory cortex, and they have a inhibitory relationship to gamma. The beta rhythms inhibit or suppress gamma, and that allows the brain to control its own processing. The feedback from the beta rhythms allows the brain, top-down control of brain, allows you to focus attention, make decisions, voluntary actions. It's a way for the brain to sculpt and control the processing and the information that's being fed to it from the outside world. So let me go just a little bit further so to be clear. Here's a study. This is the study that sort of started this line of thought. It's by Michael Lundquist. Michael Lundquist was a postdoc in the laboratory. He came and he studied working memory. He did something very unique. First of all, we had the animals trained on these tasks where they're holding two pictures in working memory. We'll call them S1 and S2. They saw two pictures in sequence, like a house and a banana. And then they had the memory delay, then house and banana. They had the answer house and banana. And when Michael did, he did something different that people hadn't done in the past. People studying working memory since 1971, when Joachim Fuster first discovered this neural correlate. And all neurophysiologists, especially in the days of single neuron recording, we did a lot of averaging across trials. We record the same condition, 20 trials, average across. And the idea is that by averaging, we're pulling out the signal and we're getting rid of the noise. But what if some of that noise wasn't actually noise? Noise is just variance that you can't explain. What if the brain's actually using that variance? So Michael looked at cortical activity in real time on individual trials. And this is what he found. Shown here on the y-axis, now we're looking at local field potentials now, power, and this is frequency on the y-axis, time on the x-axis. S1 is when the animal saw the first stimulus, short gap in time. S2 saw the second stimulus, and here's the memory delay. And the color represents the power of the local field potentials under these individual electrodes. So what did we find? Well, we found that stimulus information was carried by spiking. And it was only carried, we only saw that expressed during bursts of gamma. So we could read the contents of working memory, but only during these bursts of gamma. And you see that here, these bursts of gamma are not continuous like we thought from all the averaging over the years, but they're actually very sparse and bursty. But these bursts of gamma and the spiking carrying the sensory contents of working memory alternated with bursts of beta. And you see that here, the gamma is up here, and then the beta bursts tend to happen when the gamma bursts are not happening. And here's actually a plot. Now we are averaging across trials. We're looking at the burst rate here across time for the gamma burst rate and the beta burst rate. And what you see in gamma is the same thing that people have shown for 50 years of studying working memory. You get increases in gamma and spiking when the stimulus is seen. Then over the working memory delay, you get some sustained activity that tends to ramp up over the memory delay as the animal gets ready to read out the contents of working memory. So this profile is the exact same thing that's been seen by spiking for many, many years. But now we look at beta and look at beta, the burst rate of beta. Beta is like a mirror image of gamma. Whenever gamma goes up, beta goes down. Whenever beta goes up, gamma goes down. So beta and gamma are anti-correlated. What we did, we performed causality measures on these two signals on the beta and gamma signals. We found that beta influences gamma. Beta actually has a suppressive relationship to gamma. And through a number, and we know, everybody's known, not us, but lots of people have shown that gamma increases in gamma power associated with sensory input. And we did a series of studies where we showed that beta is actually carrying top-down information. It's not only associated with top-down information, but also associated with executive control. And I'll give you an example in a moment. But the classic example of the role of beta in control is that you record in motor cortex and you tell the subject, press a button, but wait until I say go. And the whole time they're waiting to press that button, you get high beta in frontal motor cortex. And the moment they get the go signal, beta drops, and then gamma comes up, and the person presses the button. So beta is long been known to have a suppressive role in voluntary motor actions. Well, where did cognition come from? Cognition built on our volitional motor system, our ability to produce volitional actions. That's what cognition is built on. So the same dynamics that we long been known to use for motor control, we think is also being used for top-down control. So the idea here is that the brain organizes its processing by using beta waves like a stencil. There's a theory that we published just last year called spatial computing. The idea is these beta rhythms, these beta waves traveling back and feeding back in frontal cortex, sensory cortex, they inhibit specific patches of the cortex, leaving untouched cortical networks that then could be activated by sensory inputs, carrying the working memories or producing motor outputs or carrying sensory inputs. So your brain, in other words, your brain organizes thoughts by setting the guardrails, by setting this beta stencil, then it lets reality from the outside world carried by gamma sort of fill in the details. Now, we think of the spatial computing as an update to our model with Jonathan Cohn all those years ago, only that's rather than, we used to think about top-down control as being a spiking network problem and they're all being carried by anatomy, but we now think that these top-down influences are carried by these beta waves which suppress gamma and thereby decrease spiking. So just to illustrate this, let me give you an example. Earlier, I mentioned our mixed selectivity neurons. These are neurons that have crazy combinations of things that don't seem to make sense if neurons have a single function. So I'm going to show you how spatial computing, this idea about these beta stencils, can actually explain this property of mixed selectivity. Now, I've mentioned that for a number of years before we made this discovery about beta, we thought of these mixed selective neurons as kind of being like hidden units in a neural network. They're just these middle layers that just combine things in a random way that happen to evolve. But now I think there's a principle behind it, and the principle of spatial computing. So here's how this works. Imagine a sequence memory task where the animal is holding two pictures in memory and has to remember their order. So if I show the animal padlock, butterfly, it has to answer padlock, butterfly. If I show a butterfly, padlock, it has to answer butterfly, padlock. And if you record in the prefrontal cortex during a task like this, and we have, in fact, this is the exact task that originally led to the mixed selectivity paper, what you find mixed selectivity neurons that show these nonlinear conjunctions. You'll find neurons that will activate to the padlock, but only when it's first, or only when it's second, or activate to the butterfly, or only when it's second, or only when it's first. Then you'll find even crazier neurons that you'll find a neuron that will only respond to the butterfly when it's first, but then only respond to the padlock if it's preceded by the butterfly. So every weird combination you could think of, you'll find these mixed selectivity neurons. And what this means, what mixed selectivity means, is that this population of neurons has all the information needed to solve the task. It has information about what the pictures were, stimuli the animal's holding in memory, also has information about their order, because the neural responding not just dependent on the pictures, it's also dependent on the context, whether they're first or second. So all the information is there to solve the task. So here's how we think this works. Neural selectivity to sensory inputs are basically explained by random connectivity, random patch activity coming up to the cortex. And what I have here is the same patch of cortex, and we have a bunch of random connections that result in neurons that will be activated by a butterfly. In the same patch of cortex, there's other neurons, and sometimes the same neurons that will be activated by the padlock. Okay? So what happens is that the animal is trained on this task. The animal knows it's a sequence order task. It knows something is first, something second, remember the order. And what we think is that beta is a spatially structured inhibitory influence. It essentially is a signal that says, here's the context first. This is the first stimulus. So the first stimulus is coming along. This beta signal comes down from prefrontal cortex, and it selectively inhibits a patch of cortex. Selectively inhibits a patch of cortex. Here you have a inhibitory influence here. So now only neurons on this patch of cortex can respond to the padlock. Okay, then what you have then is then the second stimulus comes along, and now the beta sets up a different pattern that deactivates or suppresses or inhibits another part of the patch of cortex. Now you have neurons here that will only respond to the butterfly when it's second. So neurons here will only respond to the first stimulus when it's first. Neurons here will only respond to the second stimulus when it's second. The same patch of cortex. So this beta, and if the stimuli appear in opposite order, the beta stays the same, but which the sensory information is what's changing. So now you have this by this spatially structured inhibitory influence of beta, you have neurons that will only respond to the butterfly when they're first, or the padlock when they're first, and vice versa for the second position, for the second stimulus, and even neurons that will respond to both in some weird combination. Okay, so that's the idea with mixed selectivity. Is that this isn't just some random piecing together of neurons like in hidden units. It's actually structured by the top-down demands, and the top-down demands determine this complex selectivity, allows the brain to make sense of this task and have all the information the brain needs to solve this task. Yeah.
For spatial attention, what feature-based attention?
This is feature-based attention in this case. This isn't spatial attention. The animal is not, there's no spatial component to this task. The animal's remembering the picture and the order, and that's it.
So the spatially structured.
By spatially structured, I mean spatially in that it's targeting a certain patch of cortex. Spatial in the brain, not spatial in the world. So these patterns of beta, and we've shown this, the different top-down demands, different rules, different concepts, different categories will produce different patterns of beta. So what the beta is doing is selectively inhibiting different patterns of cortex, back and posterior cortex, allowing these neurons to have these conjunctive properties where they're responding to both the sensory input and the top-down demand in this weird nonlinear way.
There was a question from an audience. I think this was a while ago, and I think you answered it, but let me ask the question. It says, why do we end up with synchronized waves in a large number of neurons? If this subthreshold activity is random, shouldn't it be cancel each other out? I think it's when you were talking about most of the neurons being quiet and just receiving, right? It might have been from the earlier part of the talk. So,
well, neurons are nervous system will naturally, any electrical network will naturally out. So now you have neurons that are coordinating their activity. But if you think about what's actually going on in cortex, again, most of the time, cortical neurons are not spiking. What they're doing is their membrane potentials are fluctuating, constantly fluctuating, just below the spiking threshold. So cortical neurons spend most of their time in the state where like Humpty Dumpty. They're sitting on a wall. Am I going to spike? Am I not going to spike? Am I going to spike? And all it takes is a small change in that fluctuating electric field to push them one way or another. And again, it's not random. They don't cancel each other out because these waves operate on a spatial scale that's highly organized. I'll show you an example of that organization in a moment. So when you get that kind of organization, they don't cancel one out. They reinforce one. So I propose that these gamma rhythms are carrying sensory information forward in the brain, whereas the beta rhythms are carrying top-down information in the other way, in the feedback direction. If this is true, then we should be able to see this reflected in the anatomy of cortex, because your cortex has 6 layers, as we all know. And we know from the projections from the anatomy that the superficial layers are the feed-forward projections that carry sensory information from the back of the brain to the front of the brain. And we know the deep layers of cortex are the ones that send feedback information in the opposite direction. So this gamma and beta should map onto the different layers of cortex, and in fact they do. So this is a model that shows cartoon model that shows a result of a number of experiments. And what we found is that beta in deep cortical layers is in fact stronger in deep cortical layers, is in fact carrying top-down information, and the beta in deep cortical layers projects up to superficial layers and suppresses the gamma in the superficial layers of cortex. Okay, so we found this beta gamma push pull or beta inhibiting gamma. It's taking place in the deep layers of superficial layers of cortex, as we'd expect from the feedback and feed forward anatomy. Now we did these studies originally in the prefrontal cortex, but if this is true, if this really works for executive control, it shouldn't just be in the prefrontal cortex. It should be all over cortex. It should be a common feature of cortex. So just recently, just this year, Diego Mendoza-Holiday from Bob Desmond's lab and Alex Major from my lab did a heroic series of experiments and marshaled a ton of data and looked in 14 different cortical areas in macaque monkeys. And that's what's shown here. I won't walk you through the details of the figures, but what's shown along the y-axis here is the cortical layer. 0 is the middle layer of cortex, layer 4, and the blue is the power in the alpha beta bands. And the red is the power in the gamma band. By the way, I'll switch to the alpha, beta, and gamma. Most of the power is centered on the beta band, but these boundaries between alpha, beta, someone made them up arbitrarily like 150 years ago. So it actually dips down into alpha a little bit too. So what you see here's one cortical. You see the gamma is indeed stronger in the superficial layers, and the beta is stronger in the deep layers. This is powers along the x-axis. So more beta here and more gamma here. Well, we saw this in every cortical area we looked at, 14 different cortical areas. And we also found it in different species. So that we found it in macaque monkeys, we found it in marmosets, we found it in humans using human surgical patient data, and we also found it in the mouse. So this beta, deep, gamma, superficial, which corresponds to our feedback feed-forward functions, is literally everywhere in cortex, everywhere we looked. And in fact, the effect is so robust, we can anatomically find the middle layer of cortex by finding the crossover between beta and gamma. And you actually do that online during an experiment. Okay, and we verified that using anatomy. So when something is this effect is this ubiquitous and robust, do something very important. It's something that's central to the infrastructure of cortex. Okay, so these rhythms are known. They don't just bounce all over cortex. They don't just they don't cancel each other out because they're highly organized. In fact, they form something called traveling waves. So here's an example of a traveling wave moving across the cortex. This is a electrode array, the lateral prefrontal cortex. Monkeys would be here on the side of my head. Each square is a recording electrode. There's about a 0.5 millimeter between each recording electrode, and the color indicates the voltage. And what you can see, this is actually real time. It's not averaged anyway. It's actually real raw data. And what you can see is this wave is rotating around the cortex in a counterclockwise direction. That's what it's doing. Okay, so these waves are highly organized. In fact, this is now summing data from the same subject during a working memory task. And you see that these traveling waves, they're not bouncing around like ping pong balls banging into one another. They're like the crowd doing the wave in the sports stadium. They're highly organized. They move in this particular experiment, in this particular subject, in the lateral prefrontal cortex, they move principally in two directions, back and forth in two directions. Right? So when you see organization like this in the brain, it must be having a function. In the brain, organization is function. So these traveling waves are highly organized, and in fact, they change with task demand, the cognitive demands. So here's the working memory task the subject was performing, and there's a baseline period where the animal is waiting to start the task. Here's where the animal gets the task begins. The animal starts fixating, and then it gets a picture to hold in memory. Here's the memory delay. Here's the animal made its response. So what's happening here is first during the baseline period before the task is starting, these waves are moving back and forth in a bidirectional way. And the moment the task starts, one of the directions drops out, and it becomes highly unidirectional. Highly unidirectional. Come on, pointer. There we go. Highly unidirectional until the task ends, and it goes back to this bidirectional. So these waves are highly organized, which means that these waves aren't just blasting a set of cortex with like a fire hose with beta or gamma. The fact that they're organized means they can actually have a very precise effect on excitability in networks of neurons. And in fact, we find these traveling waves primarily in these lower frequencies like beta that we associate with control. But when I say they change with task demands, what exactly are they doing? Well, we don't know yet, but we're starting to find out. Basically, we found these traveling waves correspond with shifting patterns and spiking patterns in cortex. Now, what's illustrated here is something called subspace coding. What subspace coding is, if I record from 1,000 neurons simultaneously, I can create here we're talking about spiking now. If I record from 1,000 neurons simultaneously and look at their spiking activity, I can create a 1,000-dimensional space to describe them, right? I mean, it's self-evident. Each spike rate for each neuron is along a different vector. You have a 1,000-dimensional space. But then you ask the question, how many dimensions do I actually need to explain how these neurons are spiking? And the answer is you explain it in 3 dimensions, 3 or 4 dimensions. 3 or 4 dimensions, you explain almost all the variance in the spiking patterns of neurons. And what that means is that spiking patterns in cortex, at least in this mathematical sense, can just be explained by a few dimensions. And what that means is neural spiking is kind of like birds flocking in the sky. It's not like a random crazy thing, all these spiking is happening all over the place. In the mathematical sense, in the shared common coordination sense, spiking patterns of neurons like a flock of birds moving across the sky that are changing gradually, and they're highly coordinated. Okay, so what we're finding is that these spiking patterns changing in cortex, in subspace, these shifting spiking patterns, there's a direct correspondence between traveling waves moving across electric fields moving across the surface of cortex and these shifting patterns that we see for spiking activity. When we see this rotation in subspace, this mathematical way of representing what neurons are doing, we see rotating traveling waves across cortex in the electric fields. Now, let me just mention for a moment, there's no reason in principle why this rotation in subspace is a mathematical representation, why it should correspond to a natural rotation across the surface of the brain, yet it does. And it kind of suggests that at least in some forms of brain processing are somehow mapped out in these principles in how these waves interact across cortex, as if you can as if they're like played out on a movie screen across the brain's electric fields. And this is what I mean by how these electric fields they can sculpt this activity to cause these coordinated shifting patterns we see when cortical neurons fire. Okay, so and because of effective coupling, we think that these traveling waves, these rotating or moving electric fields, are actually having a direct causative influence, actually coordinating, causing these underlying movements of spiking in the subspace. So traveling waves can help organize spiking patterns and even perform neural computation. That's the idea. And let me add one final, well, maybe not one final, but one more point about these traveling waves, electric fields. Spiking, synaptic transmission is pretty slow. It takes a while for an action potential to move down an axon. Then it takes a while for chemicals to diffuse across the synapse. Well, electric fields spread influences at the speed of light nearly instantaneously. And to my mind, spreading influences in a coordinated way at nearly the speed of light is a pretty good mechanism for coordinating activity in the brain. Okay, now for the last topic, just to bring this point home about emergent properties, is another line of investigation we're conducting in laboratories. How does general anesthesia cause loss of consciousness? Now, it's kind of been, it's maybe disturbing, but as probably many of you know, although anesthesia has been around for over 100 years, medical doctors don't really know why it makes you unconscious. There's been this tacit assumption that it kind of just shuts off your cortex. Well, it's not what happens. Oops, jumped ahead. When it's not what happens is that it actually changes the oscillatory dynamics of your cortex. So here on the left is the recordings from macaque monkey from 4 different cortical areas. This is a frontal eye fields, prefrontal cortex, so frontal cortex, parietal cortex, and STG is the auditory cortex. And these are local field potentials, and your spiking activity in real time from those same electrodes. And what you see in the awake brain, a lot of high frequency radio chatter. There's gamma waves going on, there's beta waves going on, beta waves controlling the gamma waves, there's ensembles forming, breaking apart, reforming. So you get a lot of activity. But when we anesthetize the animal with a drug called propofol, all this high frequency chatter gets replaced, gets swamped by this low frequency 1 hertz delta hum you see right here. This is you see the local field potentials just oscillating very strongly at 1 hertz, and that enframes spiking activity. Okay, so but here's the thing though, your brain also switches. This anesthetized state looks a lot like slow wave sleep, delta 1 hertz oscillations. But sleep anesthesia, although you're unconscious in both cases, they're very different. One is that anesthesia produces amnesia. You lose memories for things that happened just before you got the drug. And also, I can wake you up from sleep, but you can't wake up from anesthesia, thankfully. So what's different about sleep and anesthesia? Well, we find that this may be because anesthesia does something different than sleep. It causes these low frequency delta waves to become misaligned. There's a recent study by Alex Barton in the laboratory. She's been examining these low frequency delta waves, and what she found is that 2 different anesthetics. Before I showed you propofol, now I'm showing you results or cartoon results from 2 other drugs, ketamine and dexmedetomidine. And what they do is they, like propofol, they produce this delta slow wave kind of sleep kind of thing. But what happens is it scrambles the alignment. So cortical areas on the same side. Cortical areas There we go. There we go. The slow waves actually from the same side of the cortex, they become 180 degree out of phase of one another because of epileptic anesthesia. They become completely misaligned. And weirdly enough, signals from opposite hemispheres become more aligned, which is usually they're a little bit misaligned in the awake state. They become more aligned across hemispheres, then they become absolutely 180 degrees out of phase of one another on the same side of the cortex. Now, what's the big deal about this? Well, it could explain why anesthesia is different from sleep. But let me point out that these two different drugs we use here, and we're finding the same effects of propofol, by the way. So I'll say 3 different drugs. They act on different receptors on different neurons in different parts of the brain. Yet all 3 of these anesthetics do the exact same thing. They make you unconscious and put you in a state that allow you to recover from unconsciousness. So what is it? What are the convergence effects of these 3 anesthetics? How do they all do the same thing to your brain? Well, this will never be explained on the reductionistic level because these neurons, these anesthetics act on different neurons, they act on different receptors, different parts of the brain. There's in principle, no explanation that could unite what they're doing. But here at this more emergent level, at this higher level of functioning, at the level of these electric fields and these waves, we now have an explanation. Not a full explanation, but we have the start of an explanation on why these 3 different anesthetics do the exact same thing. We see these conversion effects at this higher level of these more emergent properties. In fact, if I would show you this slow wave data from these 3 different drugs, you could not tell them apart. They seem to do the exact same thing to the brain's electric fields. So, finally, the last thing I'll tell you about is one line of this we have is we often get asked, or there's a question about how the brain manages if neural activity are these sparse firing, these waves moving across cortex. How does the brain maintain information between the waves going by when these neurons are supposedly silent? Well, there's short-term synaptic plasticity in the brain. We've been studying that as a mechanism to help produce these waves and also help maintain information between bouts of spiking. And this is work by Leo Kotsakov and Adam Eisen laboratory. And what they found is they built a neural network model that uses this short-term synaptic plasticity, and it works just fine. It does a working memory task just fine. That's not surprising. It's how they designed how they built the model. But what they found is having this short-term synaptic plasticity, these bouts of spiking, these waves, what they do is they have all these benefits that we didn't bake into the model, but they're there. It makes neural networks function better. It makes them more robust to noise. If you take the standard model for how working memory is done, which is purely sustained activity, so-called attractor dynamic model, you take a model like that and you delete 10% of the synapses, just a small number of synapses, and the model falls apart and no longer do working memory. But you take this model that has a synaptic plasticity built in with these sparse wave-like properties, and what happens is that you can delete 40 or 50% of the synapses, and the network is still functioning. Okay, and that's a pretty good standard for good functioning networks. You don't want networks in your brain to fall apart just because you lose a few neurons. It also makes them more robust to degradation. Sorry, I said that first. Degradation makes them more robust to noise, that the networks can process noisy stimuli more effectively with these dynamics than it can, which is purely attractor dynamics. And finally, it also helps stabilize networks so that neural activity doesn't run wild. This is actually a real problem in the brain because your brain actually operates on a knife's edge. It has to be excitable enough so that its parts can influence one another, but if it gets too excited, then it goes into epileptic activity. And so the brain operates on this really knife's edge of excitability, just enough excitability and not too much. And our lab's identifying a number of different mechanisms that allows the brain to operate within this very narrow excitability range. And this short-term synaptic plasticity is one of them. So, conclusion, the neural activity underlying cognition is not smooth or steady state like we used to think, like I used to think. It's complex, dynamic, and rhythmic. There's emergent properties like traveling waves. And this is providing new ones, insights into mechanisms and functions. More specifically, take-home message is that lower frequencies, especially alpha beta waves, are the top-down control signals, and they suppress, they inhibit the higher frequency gamma waves and spiking that carry the context of thoughts and also produce motor outputs. I talk mainly focused on sensory inputs. And this is a cortical motif that we see everywhere we look in cortex. We've seen across 4 different species. And it's just to play this motif, this beta gamma interactions play a general role in controlling cortical activity, not just working memory, but working memory, attention, predictive coding, basically anything in the cortex. So the cortex has to constantly balance the flood of information coming into the brain and produce the kind of organization that needs to manage that sensory information, not get overloaded, and do something goal-directed with it. So I thank you for your attention. I thank you for listening to me talk.